Now we will study about biotechnology. At its core, biotechnology is simply using living tools to make things that help humans. Humans have actually been biotechnologists for thousands of years without calling it that. Every time we use bacteria to turn milk into curd, yeast to make bread, or fermentation to create wine, we are using live organisms to create a product. While making bread is technically biotech, today the word usually implies something more high tech. Modern biotechnology focuses on genetically modified organisms. Instead of just using a microbe as is, we tweak its DNA to make it work better, faster, or produce something entirely new on a massive scale. Key examples of modern biotechnology includes 1. In vitro fertilization, where life begins in a lab. It is also called as test tube baby process. 2. Gene synthesis, which includes creating a specific piece of DNA from scratch. 3. DNA vaccines, using genetic material to teach our immune system how to fight diseases. 4. Gene therapy, which involves fixing or replacing a broken gene inside a person's body to cure a disease. To make sure everyone is on the same page, the European Federation of Biotechnology created a definition that blends the old and the new. The integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services is the definition of biotechnology. Let us break down this definition. Natural science plus organisms means combining our knowledge of biology with the actual living thing. Cells and parts thereof means we don't always need the whole animal or plant. Sometimes we just use one cell or one enzyme. Molecular analogues means these are man made molecules that mimic natural ones to get a specific job done. Products and services means the end goal could be a life saving medicine, which is a product, or way to clean up oil spills, which is a service. Now let us study about principles of biotechnology. Modern biotechnology is not just one thing. It is built on two core techniques. First is genetic engineering. This is the editing phase. We are literally altering the chemistry of DNA or RNA and putting it into a new organism to change how that organism looks or functions. Here, genotype is changed to see changes in phenotype. Second is bioprocess engineering. This is the factory phase. Once we have our engineered cell, we need to grow it in massive quantities. This requires a perfectly sterile, germ-free environment so that we only grow the good microbes used for making things like vaccines, enzymes, and antibiotics. In the past, we used traditional hybridization or breeding. Think of a farmer breeding two types of corn. What is the problem? It is messy. You get the good genes you wanted, but you also get a bunch of undesirable genes you did not want. Genetic engineering is like using a sniper rifle instead of shotgun. It allows us to isolate and move only the specific gene we want, leaving the junk behind. If you just drop a random piece of alien DNA into a cell, the cell will likely ignore it or destroy it. To make the DNA stay and multiply, it needs to join a chromosome at a very specific spot called the origin of replication. Think of the origin of replication as a start button. Once the alien DNA is linked to this button, the cell recognizes it as its own and begins making copies of it every time it divides. This process of making identical copies is what we called cloning. In 1972, two scientists, Stanley Cohen and Herbert Boyer, changed history. They wanted to see if they could move an antibiotic resistant gene from one bacteria, that is from Salmonella, to another bacteria, that is E. coli. They used three specific tools that every biotech student needs to know. First is restriction enzymes. These are the molecular scissors that cut DNA at exact locations. Second is plasmids. These are small circular rings of DNA. In this story, the plasmid acts as a vector or a delivery truck to carry the gene into the new cell. Third is the DNA ligase. This is the molecular glue that joins the cut DNA ends together. By cutting the gene, gluing it into a plasmid, and putting that plasmid into E. coli, they created the first recombinant DNA. If you want to genetically modify an organism, you always follow these three basic steps. First is identification. Find the DNA that has the gene you actually want. Second is introduction. Get that specific DNA into the host organism. Ensure that the DNA stays in the host and gets passed down to all its children. Now let us see about tools of recombinant DNA technology. First is restriction enzymes. Imagine you are a biological architect. To remodel DNA, you can't use regular tools. You need microscopic ones. The primary tools in our kit are restriction enzymes, which are like the scissors, polymerases, which are like the photocopy machines, ligases, which are like the glue, vectors, which are like the delivery truck, and host organisms, which are like the factory. In 1963, scientists found that certain bacteria could cut up the DNA of invading viruses, which are bacteriophages, to stop them from growing. The enzymes doing the cutting are called restriction endonucleases. We use a specific code to name these enzymes. Let's look at EcoRI as an example. Here, E, the first letter comes from the genus Escherichia. Co, the next two letters come from the species coli. R, this is the name of the specific strain. Here it is RY13. I, the Roman numeral shows the order in which it was discovered in that bacteria. There are two types of cutters, exonucleases and endonucleases. Exonucleases cut DNA at very ends. Endonucleases may cut at specific point inside the DNA strand. Restriction enzymes don't just cut anywhere. They look for a specific address called a recognition sequence. These sequences are palindromes. In English, a palindrome is a word like madam. It reads the same word forward and backward. In DNA, a palindrome means the sequence reads the same on both strands when you read in the same direction, that is 5' to 3'. Example, if top strand reads from 5' to 3' as GAATTC, bottom strand also reads as GAATTC from opposite side. When the enzyme cuts, it doesn't usually make a straight blunt cut. It cuts a little off center. This leaves short single-stranded overhangs called sticky ends. They are called sticky because they want to form hydrogen bonds with their matching partners. If you cut two different DNA samples, like a human genome and a bacterial plasmid, with the same enzyme, their sticky ends will match perfectly. DNA ligase then acts as the glue to join them together forever. Once you have cut the DNA, you end up with a soup of different sized fragments. To separate them, we use a technique called gel electrophoresis. Let us see how it works. DNA is naturally negatively charged. We place the DNA in a gel made up of agarose, which obtained from seaweed, and apply an electric current to this. The DNA moves away from the negative end towards the positive end, that is anode. The gel forms sieve. It acts like a thick forest. Smaller fragments move through the gaps easily and travel farther, while larger fragments get stuck and stay closer to the start. DNA is invisible to the naked eye. To see it, we stain the gel with a chemical called ethidium bromide. Then shine UV light on it. The DNA fragments will glow as bright orange bands. Now, the final step is elution. Once we identify the band we want, we literally cut that piece of gel out and extract the DNA. This process of extracting the DNA from the gel is called elution. Now, that purified DNA is ready to be joined with a vector to create a recombinant molecule. Next, we will talk about cloning vectors. To understand biotechnology, you first need to understand the delivery truck of the molecular world. In nature, certain entities like plasmids, which are small circular DNA in bacteria, and bacteriophages, which are viruses that infect bacteria, have a special power. They can replicate inside a host cell independently. They do not wait for the cell's main chromosome to tell them what to do. They just start making copies of themselves. Some plasmids only make one or two copies, but others are like high speed photocopiers making 15 to 100 copies or even more. If we hitch a piece of foreign DNA, that is the DNA we want to study, onto these vectors, that foreign DNA gets copied just as many times. A vector should have three specific features. First is origin of replication. This is a specific DNA sequence where replication begins. If you link your DNA to the ORI or ORI, it will get replicated. Crucially, the origin of replication also controls the copy number. If you want thousands of copies of your gene, you pick a vector with an origin of replication that supports high speed replication. Second feature of vector is selectable marker. When we try to put DNA into bacteria, a process called transformation occurs. Not every bacterium will take it up. We need a way to find the winners. We use genes for antibiotic resistance like ampicillin or tetracyclines as markers. If a bacterium successfully takes up the vector, it survives the antibiotic. If it does not, it dies. This allows us to select only the transformed cells. Third feature is cloning sites. To insert our foreign DNA, we need a specific spot to cut the vector open. We use restriction enzymes for this. A vector should ideally have only one recognition site for an enzyme. If it has many, the vector will be chopped into many pieces, making it useless. We usually insert the foreign DNA right into the middle of an antibiotic resistance gene. This breaks that specific gene. A process called insertional inactivation occurs. Now, how do we know if our DNA actually got inside the vector? We use two main methods. The old way was by antibiotic selection. See, if we insert our DNA into the tetracycline gene of the plasmid PBR322, the plasmid loses its ability to survive tetracycline. We then grow the bacteria on ampicillin. We take the bacteria which survived in ampicillin and then we plate them on tetracycline. The ones that die on the tetracycline are the ones that actually have our foreign DNA. What happened here? Insertional inactivation occurred. The new way is by blue-white screening because double plating is tedious. Scientists developed a chromogenic substrate. We insert the DNA into a gene called beta-galactosidase. If no insertion occurs, the gene works and the colony turns blue. When gene gets inserted into beta-galactosidase, the gene is inactivated and the colony stays white. White colonies are our winners. That is, they have a gene of interest. Finally, we have learned how to disarm natural enemies to help us. For plants, we use Agrobacterium tumefaciens. In nature, it gives plants tumors using a T plasmid. We have modified this plasmid so it no longer causes disease but still acts as a master at delivering genes into plant cells. For animals, we use retroviruses. Normally, these turn healthy cells into cancerous ones. Scientists have disarmed them so they can deliver helpful genes into animal cells instead. Once you loaded your DNA into the right vector, it is a just matter of parking that vector in a host cell and letting the natural machinery of life to do the copying for you. Now let us talk about competent host. The question is, how do we actually get recombinant DNA inside a living cell? It is harder than it sounds. You see, DNA is a hydrophilic molecule. In simple terms, it loves water but hates fats. Because cell membranes are made up of fats, they act like a waterproof barrier. DNA simply cannot pass through them on its own. To solve this, we have to make the host cell competent. Think of this as a preparing the cell to be willing or able to accept the foreign DNA. The most common way we do this with bacteria is through a chemical and temperature based process. It happens in three specific steps. Step 1 is the chemical soak. We treat the bacteria with a specific concentration of a divalent cation like calcium. This chemical treatment opens up tiny pores in the bacterial cell wall, making it easier for DNA to get close. Step 2 is the chill. We incubate the cells and recombinant DNA together on the ice. Step 3 is the shock. We suddenly move them to a temperature of 42 degrees Celsius for a brief moment. This is the heat shock and then immediately plunge them back onto ice. This rapid change in temperature forces the bacterial cell to gulp down the recombinant DNA through those pores. Now, while heat shock works great for bacteria, other types of cells require more specialized delivery tools. If we take an animal cells, here micro injection is used. Imagine a needle so tiny it can pierce a single cell. In micro injection, we take the recombinant DNA and inject it directly into the nucleus of an animal cell. It's precision work at its finest. Next is for plant cells, we use gene gun or biolistics. Plant cells have tough cell walls, so we use a more aggressive approach called biolistics. We coat microscopic particles of gold or tungsten with DNA and literally fire them into plant cells at very high velocity. It is essentially a DNA shotgun. Finally, we can use nature's own experts like viruses and bacteria. We take a pathogen, disarm it so it can no longer cause disease, and then load it with our recombinant DNA. When we allow this disarmed pathogen to infect the host, it naturally injects our desired DNA into the cell for us. So, whether we are using temperature shocks, tiny needles, gold bullets, or tamed viruses, the goal is always the same: overcoming the cell's natural defenses to get our genetic blueprint inside. Now that we have mastered the tools for constructing and delivering recombinant DNA, let's move on to the actual processes that make this technology work in a lab setting. Now, let us discuss about processes of recombinant DNA technology. To build something new like a bacteria that produces human insulin, we have to follow a very specific sequential steps. The full process involves seven major steps. First is isolation of the DNA. Second is fragmentation using molecular scissors. Third is selection of the desired fragment. Fourth is the ligation into a vector. Fifth is the transformation, that is, moving it into the host. Sixth is culturing the host at a large scale. And seventh is extraction of the final byproduct. Before we can manipulate a gene, we have to get it out of the cell. In almost every living organism, the blueprint for life is DNA. But there is a catch. DNA does not just float around freely. It is locked inside a fortress of cell walls and membranes, and it is tangled up with other molecules like RNA, proteins, and lipids. To use restriction enzymes later to cut the DNA, that DNA must be pure. Now, our goal is to strip everything away until we have nothing left but the DNA threads only. Our first task is to break the cell open. Depending on what organism we are working with, we use specific enzymatic keys to dissolve the outer layers. For bacteria, we can use an enzyme called lysozyme. For plant cells, we use a cellulase to break down the tough cellulose wall. For fungi, we use chitinase. Once the wall is breached, the cell releases its contents, which is a messy soup of DNA, RNA, proteins, and fats. Now we have a mixture, but we only want the DNA. We need to systematically destroy the contaminants using specific enzymes. We add ribonuclease. This, this digests the RNA into tiny pieces. DNA is often wrapped around proteins called histones. We use proteases to chew up these proteins and set the DNA free. Other treatments are used to clear out polysaccharides and lipids. At this stage, the DNA is dissolved in solution and you can't see it yet. To make it visible and physical, we use a classic lab trick called chilled ethanol. DNA does not dissolve in cold alcohol. When we add the chilled ethanol, the purified DNA suddenly precipitates or crashes out of the liquid. If you were looking at the test tube right now, you would see it appear as a collection of fine white threads suspended in the liquid. This is the raw material ready for the next step of engineering. Next is cutting of DNA at specific locations. Once we have our purified DNA, we need to cut it. We do not use physical blades. Of course, we use restriction enzymes. Think of these enzymes as highly programmed molecule scissors. They don't just cut anywhere, they look for a very specific code or sequence to make their snip. We put the DNA and enzymes together in a tube and keep them at the optimal temperature and pH that the enzyme likes. If the conditions are not perfect, the scissors won't cut. How do we know if the cutting worked? We cannot see DNA with the naked eye, so we use a technique called agarose gel electrophoresis. Imagine a slab of gel with a positive electric charge at one end and a negative charge at the other. DNA molecules are naturally negatively charged. When we turn on the electricity, the DNA tries to run away from the negative side and move towards the positive electrode called the anode. Smaller pieces of DNA move through the gel much faster than the big ones. By looking at how the DNA fragments have spread out, we can confirm if our scissors did their job correctly. While we are cutting our gene of interest from the source DNA, we have to do this exact same thing to our vector DNA. The vector is usually a small circular piece of DNA, like a plasmid, that will carry our gene into the host cell. We use the same restriction enzyme to cut the vector. This ensures that the cut on the vector matches the cut on our gene like two matching pieces of jigsaw puzzle. Now comes the final assembly. We take our cutout gene of interest and our opened up vector and mix them together in a tube. To make them stick permanently, we add a special enzyme called ligase. If the restriction enzymes were the scissor, DNA ligase is the glue. The ligase seals the backbones of the two DNA molecules together. The result is a brand new hybrid molecule known as recombinant DNA. We have officially recombined DNA from two different sources into one single strand. Let us discuss about amplification of gene of interest using PCR. Where does PCR fits? It is done to increase copy number of DNA of interest before it is ligated into vector. Think of this as the molecular photocopier. What is PCR? PCR stands for polymerase chain reaction. It is a technique used to take a single segment of DNA and flip the copy switch until you have millions or even 1 billion copies, all within a small test tube. We call this process amplification. To run this photocopier, we need a specific chemical soup. First is the template. This is the segment of DNA you want to copy. Second is primers. These are short chemically synthesized pieces of DNA. Think of them as a start here signs. They attach to specific ends of the gene you want to copy so the enzyme knows where to begin. Third is nucleotides. These are the raw building blocks like ATGC that will be used to build the new DNA strands. ATGC are adenine, thymine, guanine, and cytosine. Now let us talk about the enzyme Taq polymerase. The most important ingredient for the PCR is the enzyme DNA polymerase, which does the actual building of the copies. But there is a catch. To copy DNA, we first have to unzip the double helix using extreme heat, a process called denaturation. Most enzymes, like the ones in our body, would melt and stop working at these high temperatures. To solve this, scientists use a special thermostable enzyme called Taq polymerase. This enzyme is isolated from a bacterium called Thermus aquaticus, which lives in the boiling hot springs. Because it is a heat lover, it stays perfectly active even when the DNA is being heated up to unzip. It is the engine that makes the whole reaction possible. The beauty of PCR is that it is a cycle. We heat it up to unzip, we cool it down so the primers can stick, the Taq polymerase builds the new strand. Then we repeat. If you repeat this cycle many times, the DNA grows exponentially. After about 30 cycles, you have gone from one single segment to 1 billion identical copies. At this point, you have a massive concentration of our gene. You can now take these amplified fragments and move to the next step we discussed, that is, ligating them into a vector for further cloning. Now we have already seen that gene of interest is added to the vector. That is, we have built our recombinant DNA and we have made millions of copies of it. But right now, it is just sitting in a test tube. To make it come alive and actually produce proteins, we need to put it into a living factory, usually a bacterium like E. coli. Getting DNA into cell is harder than it sounds. Cell membranes are like high security gates. They don't just let large foreign molecules enter in. Before we can insert our DNA, we have to make the recipient cells competent. This is a laboratory treatment, often using chemicals like calcium chloride or heat shock that temporarily stresses the cell, forcing its pores to open just enough to let the DNA in from the surrounding liquid. When a cell successfully takes up that foreign DNA and begins to host it, we call that process transformation. But here is the catch again. Transformation is not 100% efficient. In a tube of million bacteria, maybe only a few hundred will actually take up the recombinant DNA. The rest stay exactly as they were. This leads us to big question, how do we find the transformed cells in a crowd of millions? To find our transformed cells, we use a clever biological trick involving antibiotic resistance. Imagine the vector we have built earlier did not just have our gene of interest, it also contained a survival gene, specifically a gene for ampicillin resistance. Here is how the selection works. We take all our bacteria, the ones that took the DNA and the ones that did not, and spread them on a petri dish. The dish contains agar mixed with ampicillin, a powerful antibiotic that usually kills E. coli. The untransformed cells, that is, the ones that fail to take up our DNA, have no protection. They land on the plate and die. Only the cells that successfully took up our recombinant DNA have the resistance gene. They survive, grow, and multiply into visible clumps called colonies. In this scenario, that ampicillin resistance gene is called a selectable marker. It is called a marker because it marks the successful cells, and selectable because it allows us to select the winners while the others are eliminated. Without a selectable marker, finding the one in a million cell that took our DNA would be like finding a needle in a haystack. With it, the haystack simply disappears, leaving only the needles behind. Now, next we will see about obtaining the foreign gene product. This stage is production phase, where we move from the molecular level to the industrial level. The ultimate aim of almost all recombinant DNA technology is to produce a protein. When you insert alien DNA into host cell, that cell starts multiplying. And if the conditions are right, it starts reading that new DNA like an instruction manual. When a host cell, like a bacterium, produces a protein based on a gene from a different organism, we call that a recombinant protein. The host itself is called a heterologous host because it is hosting a gene that does not naturally belonging to it. In the beginning, we grow these cells in small flasks in a lab. This is great for testing, but a small flask doesn't produce enough protein to treat thousands of patients or supply a whole country. To go big, we have to scale up. But you cannot just get a bigger bucket. You need a system that keeps the cells in their happiest, most productive state. In biology, we call this the log phase or exponential phase. This is the stage where cells are young, healthy, and multiplying at their maximum speed. One way to keep cells in that high energy log phase is a continuous culture system. Instead of letting the cells sit in their own waste, we do two things simultaneously. We drain out the used up medium from one side and we pump in a fresh medium from the other side. By keeping the environment fresh, the cells never get tired or run out of food, which leads to a much higher yield of our desired protein. When we talk about large scale, we mean bioreactors. These are massive vessels that can hold anywhere from 100 to 1000 liters. Think of a bioreactor as a giant high tech womb. It provides the perfect conditions for the cells to thrive, including optimal temperature and pH, plenty of oxygen, the right mix of salts, vitamins, and substrates. The most common type of bioreactor is the stirred tank reactor. If you look inside one, you would see it is usually a large cylinder with a curved base. Curved base is to make sure nothing gets stuck in the corners. It is packed with systems to keep the soup perfect. It has 1. The agitator. It is a stirrer that keeps everything mixed so the food and oxygen are evenly distributed. 2. Oxygen delivery. Sometimes air is bubbled through the bottom so the cells can breathe. 3. The control centers. There is a sensors that monitor and automatically adjust the temperature and pH. 4. Foam control. Since biology can get messy and bubbly, there is a system to break down foam so it does not overflow. 5. Sampling ports. These are little trapdoors that allow scientists to pull out a small amount of the culture to check on it without stopping the whole machine. Now we will see about the downstream processing. But right now, your desired protein is floating in a giant tank mixed with billions of bacteria, dead cells, leftover food, and metabolic waste. You can't just bottle that and give it to a patient. This final critical phase is called downstream processing. What is downstream processing? If the bioreactor is the upstream part where we grow the product, downstream processing is everything that happens after the harvest to turn that raw biological soup into a. It consists primarily of separation and purification. First, we have to isolate the protein. Separation. We use techniques like filtration or centrifugation to physically separate the cells from the liquid. If the protein is inside the cells, we have to break them open. If it is secreted into the liquid, we harvest the fluid. Next is purification. This is the most delicate part. We use highly specific methods like chromatography to pull our specific protein away from all other similar looking proteins until it is 99.9% pure. At this stage, the alien DNA product is finally on its own. Once we have the pure protein, we cannot just put it in a vial of water. Most proteins are fragile and will break down quickly. We have to create a formulation. This means mixing the protein with specialized preservatives and buffer solutions that keep it stable, active, and safe for storage on pharmacy shelf or in a hospital refrigerator. Before a single dose is sold, it must undergo strict quality control testing. Every single batch is tested to ensure it is at the correct concentration, it is completely sterile without any bacteria, and it is chemically pure. If the product is a drug like insulin or vaccine, it must also go through rigorous clinical trials. This involves testing the product in living organisms and humans to prove that it actually works and, most importantly, that it is safe. One thing to remember is that downstream processing is not one size fits all. The way you purify a vaccine is very different from how you purify an enzyme used in laundry detergent. The steps, the quality tests, and the regulations vary from product to product depending on how the product will be used.